Okay, let's unpack this. Today, we are undertaking a deep dive into the molecular genetic techniques that really, they form the absolute foundation of modern cell biology.
They really do.
If you want to understand how a cell works, you have to be able to talk to its genes.
That's the crux of it. I mean, the central challenge in our field isn't just watching what cells do, it's defining that behavior in terms of, exact chemical and molecular mechanisms.
Right.
And to get to that level of resolution, you almost always have to manipulate the fundamental blueprint, the gene that encodes the protein you're interested in.
So our mission today is to explore that toolkit, the one that lets researchers answer those huge foundational questions we're always asking, like what is the precise function of a protein inside a living cell?
Where is it located exactly?
When is it expressed?
The-
Developmentally, cyclically.
And maybe the most fascinating one, how did this whole system evolve across species?
It's compelling right from the start. You look at that foundational diagram, figure 6.1, and you see that the whole discovery process is this loop of convergence and divergence.
It is. You might start with a specific observation, say a mutant organism with a weird trait.
Or a protein you purified in a test tube that does something interesting.
Right, or even just a sequence you found in a massive genome database. But no matter where you start, the goal is always to isolate and clone the gene behind it.
And once you have that cloned gene,
Yeah.
boom, the paths just open up again.
Totally. You can use it to figure out its evolutionary history by comparing sequences. You can determine its exact location by, tagging the protein it makes.
Or you can mass produce the protein for structural studies.
Mhm.
Or, and this is crucial, engineer new mutant organisms to study what happens.
It's an iterative process. Genetics and biochemistry just constantly feed back on each other.
We see this perfectly in that classic story of NSF and Sec18. It just bridges that historical gap between the two fields so beautifully.
Oh, it's the perfect example.
So walk us through that. How did two completely separate research paths end up proving they were studying the exact same thing?
Okay, so on one side, you've got the biochemists. They're focused on mammalian cells, and they're purifying a protein based purely on what it does in a test tube.
Okay.
Specifically, its sensitivity to a chemical, N-ethylmelamide, that's where it got its name, NSF, N-ethylmelamide sensitive fusion protein.
So a purely chemical definition.
Purely chemical. They defined it by its ability to help cellular vesicles fuse with their target membranes. That's all they knew.
And at the same time, you have geneticists working in a completely different world, Baker's yeast.
Right. Saccharomyces cerevisiae, the ideal model. The geneticists are doing these big systematic screens to find mutations that mess up protein secretion. They call them Sec mutants.
For secretion defect.
Exactly. And they find a gene they call Sec18. When you mutate Sec18, the yeast cell gets this massive traffic jam of vesicles that can't go anywhere.
So a clear defect in vesicle trafficking. A genetic definition based on a visible problem.
Precisely. So you have one group with a protein defined by its mechanism fusion, and another with a gene defined by its functional defect trafficking.
And the big moment of convergence.
It happened when the gene for the yeast Sec18 mutation was finally sequenced. Scientists found that its amino acid sequence was incredibly similar, homologous, to the sequence of that mammalian NSF protein.
Wow.
It proved that NSF and the Sec18 protein were the same fundamental conserved machine governing vesicle fusion all the way from yeast to humans. The function and the genetic defect just snapped together perfectly.
What's so remarkable is that whole process, purification, screening, sequencing, that took years. The modern landscape, because we have the genome sequenced, it just looks totally different.
Oh, absolutely. Today, we don't have to wait for that slow, painstaking convergence because we have the complete genomic sequences for humans, mice, flies, yeast, you name it.
So the pace is just...
exponentially faster. If you purify just a tiny piece of an unknown protein today, you can instantly search the genome, find its gene, and within seconds, compare that gene to every known sequence in existence. It gives you instant clues about function and evolution. It just bypasses decades of old school work.
That brings us perfectly to the first major pillar of this work, using genetic analysis of mutations. The power of genetics is really rooted in control, isn't it?
It is. Making a precise, controlled change in a gene and then just observing the effect on the organism, the phenotype.
And that approach reveals so much.
It reveals three crucial layers. First, it tells you which genes are essential for a process. Second, by combining mutations, it can reveal the exact order proteins act in a pathway.
And third.
And third, it can tell you if and how those proteins physically interact or maybe compensate for each other.
Okay, before we dive in, a quick note on terminology, though I know our audience is pretty familiar. We use alleles for the different versions of a gene. A mutation is often a recent induced change, maybe from a mutagen like EMS.
Right, ethylmethane sulfonate. It causes a heritable change in the DNA. And we track the genotype, the specific set of alleles an organism has, and relate that to the phenotype, which is the observable trait.
And our baseline is always the wild type, the standard non-mutant reference.
Yeah, and the important thing here isn't just defining the terms, it's understanding why the distinction between different alleles like recessive versus dominant gives you immediate clues about function.
Exactly. And how a mutation shows up depends a lot on the organism's ploidy. Most complex life, like us, we're diploid. We have 2 copies of each chromosome.
But many simple eukaryotes are haploid. And yeast, Saccharomyces cerevisiae, is just indispensable scientifically because it can exist stably in both states. It's incredibly versatile.
So if we focus on diploid organisms, like us, a mutant allele is classified as recessive if the mutant phenotype only shows up when the individual is homozygous.
Meaning both copies of the gene are mutated.
Right. And what does that tell us about what the protein is actually doing?
Recessive mutations are, overwhelmingly the result of a loss of function. The mutation just inactivates the gene or makes a non-functional protein.
And since one good wild-type copy is usually enough to do the job.
Exactly. One good copy makes enough protein for a normal phenotype, so you have to knock out both copies to see the defect. It confirms the gene is necessary, but that one good copy is enough.
But on the other hand, you have a dominant allele, which only needs one mutant copy. It shows you the phenotype, even if you're heterozygous, one mutant, one wild type.
And dominant alleles are usually associated with a gain of function.
What does it mean in practice?
It could mean the protein is just hyperactive, or it's being expressed at the wrong time or in the wrong place, or it's even acquired a totally new harmful function.
But the exceptions are often where the really deep insights are, especially those dominant mutations that are still fundamentally a loss of function.
Yes, 2 key exceptions. The first is haploinsufficiency. This is when one good copy of the gene just can't produce enough protein product for normal function.
Like an enzyme in a really busy metabolic pathway.
Perfect example. You remove one copy, the total enzyme level drops by half, and that drop is enough to cause the mutant phenotype. So the loss of function looks dominant.
And the second one is really fascinating, the dominant negative mutation.
That's where the mutant protein actively gets in the way of the wild-type protein. It interferes with it.
Wow.
It most often happens when the protein normally works as part of a complex, like a dimer or something. If the mutant subunit gets into the complex, it's like a bad apple. It poisons the whole thing, disrupting its assembly or activity, even though there's still good protein being made.
That distinction loss versus gain of function, and especially dominant negative, that has to be fundamental for designing drugs, right?
Absolutely. If a disease is dominant negative, you have to find a way to block the mutant protein. If it's haploinsufficient, maybe you just need to boost the expression of the good copy you still have.
And we also have to remember that dominance or recessiveness, it's all defined relative to the specific trait you're looking at. The sickle cell allele, HbS, is the classic case.
It's a perfect demonstration. If the trait you're measuring is severe anemia, the HbAs allele is recessive. You need 2 copies to get the debilitating disease.
But if the trait you're measuring is resistance to malaria,
then the HP's allele is dominant. Heterozygous individuals with 1 normal and 1 sickle cell allele are significantly protected from severe malaria. The same allele behaves totally differently depending on the biological context.
now what if the gene you want to study is absolutely essential for life, a so-called housekeeping gene? If you knock it out completely, the organism just dies and you can't study it.
That's where conditional mutations are essential. These are like genetic time bombs. They only show their phenotype under specific, non-permissive conditions.
And the most common type is the temperature-sensitive mutation.
Right. The genius of this system is that at a cooler, permissive temperature, say 23 degrees C for yeast, the mutant protein is just stable enough to fold correctly and function. The organism grows fine.
But you crank up the heat.
You crank it up to the non-permissive temperature, maybe 36 degrees C, and that subtle mutation causes the protein to rapidly unfold, denature, and lose function. And suddenly, the defect is revealed.
This was the strategy Leland Hartwell used to identify dozens of genes critical for the yeast cell division cycle, the CDC mutants.
It was a brilliantly designed screen. Because it wasn't enough to just find cells that couldn't grow at the high temperature, they had to prove the defect was specific to cell division.
So tell us about that CDC screen, which is in Figure 6.6. Why was the observation of uniform arrest the key insight.
Okay, so Hartwell's team mutagenized the yeast and grew colonies at the permissive temperature. Then they replica plated them onto a new plate at 36 degrees C to find the ones that failed to grow.
The temperature-sensitive candidates.
Exactly, but the critical step was taking those candidates and looking at them under a microscope after shifting them to 36 degrees C for a few hours.
Because a general metabolic problem would just make the cells sick and they'd stop growing randomly.
Precisely. They'd look irregular or rested all over the place. But the truly important CDC mutants, they all arrested at a single uniform stage of the cell cycle.
For example,
For instance, the CDC28 mutant arrested completely before DNA synthesis even started, before a bud emerged. But the CDC7 mutant arrested much later with a big, fully formed bud just before separation.
so that uniformity proved the defective gene was required for a specific sequential step.
It let them freeze the molecular action right in the middle of the process.
Okay, so once you've collected, say, 100 different CDC mutations that all show the same phenotype, they all arrest with a large bud.
Yeah.
How do you know if you've hit one gene 100 times? Or 20 different genes that all block the pathway at that same step?
That's when you need complementation analysis. It's the technique for figuring out how many distinct genes are behind a single phenotype. It's laid out in figure 6-7.
And it works because most of these mutations are recessive.
Exactly. So the method relies on the fact that a wild-type allele can supply the missing function. You take two recessive haploid mutants, let's call them mutant X and mutant Y, that both have the same large bud defect. You mate them.
To create a diploid heterozygote XY.
Right. And if that resulting diploid is wild-type, it grows just fine at 36 degrees C. We say the mutations complement each other.
Meaning they're in different genes.
They have to be. The wild-type gene X from the Y parent fixes the defect in X, and the wild-type gene Y from the X parent fixes the defect in Y. Each partner provides what the other one lacks.
But if the diploid is still mutant, it still fails to grow at 36 degrees C.
Then the mutations fail to complement. They must be in the same gene. The diploid is still effectively homozygous for the defect. Artwell's team used this to organize hundreds of mutations into about 20 distinct CDC genes.
So complementation tells you how many unique genes you have. The next trick, double mutant analysis, tells you the order they function in.
This is an incredibly powerful deductive tool. If two mutations cause different but related defects, the phenotype of the double mutant often just solves the puzzle of their functional sequence.
Let's stick with the secretory pathway, the Sec mutants as an example. It's a nice linear pathway.
Okay, so the pathway is ER to Golgi to secretory vesicles to the plasma membrane.
Right.
Let's say we have Sec12, which at high temperature causes the cell to fill up with ER-like membranes. And we have Sec4, which causes an accumulation of secretory vesicles, a later stage.
So we create the double mutant, Sec12, Sec4, what happens?
The double mutant accumulates ER membranes. It looks identical to the Sec12 single mutant. And the rule from this, as shown in figure 6-8, is that the double mutant always shows the phenotype of the gene that acts earlier in the pathway.
So because the block at step A, Sec12, happens before the block at step B, everything just piles up at step A.
You got it. It was a conclusive way to map the topological order of the secretory pathway.
How does this same logic work for signaling pathways? You're not tracking physical vesicles piling up, you're tracking information flow.
The logic is a bit more complex, but the principle is the same. You usually need two mutations that have opposite effects on some measurable output, like a reporter gene.
Okay, so mutation A turns the reporter off, and mutation B makes it stuck on.
Perfect. You build the double mutant, and whichever phenotype wins out tells you the order. If the double mutant is off, like A alone, it means A's action is downstream of B's. A is the final switch, and it can override B.
And if the double mutant is stuck on, like B alone, then B must be downstream of A.
Exactly. It lets you distinguish between all the different possible regulatory schemes. Does A inhibit B? Does B activate A? The double mutant gives you the answer.
Beyond just ordering pathways, double mutants are also key for revealing how proteins are related structurally or functionally through things like suppression and synthetic lethality.
Yeah, genetic suppression is this really surprising result. It's laid out in figure 6-9A. You have a mutation in gene A that causes a defect, and a mutation in gene B that causes a defect, but the double mutant, AB, is suddenly wild type again. The defect is suppressed.
Wait, how does breaking two things sometimes fix the problem?
It implies a compensatory structural change. It tells you the two proteins probably interact. The first mutation, A, breaks that interaction. The The second mutation, B, then causes a subtle change in the other protein that, just happens to restore the fit.
Like changing the lock to fit the new broken key.
That's a great analogy. It was early in vivo evidence that proteins like yeast actin and SAC6 physically touch each other.
And the opposite of that, synthetic lethality, is often the most insightful result from modern genomics.
Oh, absolutely. Synthetic lethality is when the double mutant is dramatically sicker than either single mutant, often just dead.
And this can happen for a couple of reasons, right?
Right. Case 1, as shown in Figure 6.9b, is a direct interaction failure. Partial defects in two proteins that work together become a complete breakdown when you combine them.
But case 2, functional redundancy, seems like the really critical insight for complex genomes like ours.
It is. In redundancy, you have two distinct non-essential pathways, A and B, that can both produce the same essential product. If you mutate A, it's fine because B takes over.
Mutate B, fine, A takes over.
But if you mutate both, as in Figure 6.9c, the essential product can't be made, and the double mutant is lethal. This is so powerful for finding genes whose functions are duplicated or overlap. They look non-essential in single mutant screens, but they're crucial for the cell's robustness.
And this kind of high-level genetic analysis has now been scaled up to an almost industrial level. We're talking about mapping entire cellular networks based purely on these interactions.
That's the beauty of the yeast genetic interaction map in Figure 6.10. It was this massive, systematic project to create almost every possible double mutant among the 6,000 yeast genes.
And a computer then analyzed the data.
Right. Algorithms assess the strength and type of genetic interaction for every single pair. Did combining them make the cell sicker, healthier, or kill it?
So this map is based purely on phenotype. No biochemistry was needed to get started.
Correct. And the amazing conclusion is that when you map the results, genes involved in related processes, like vesicle traffic or cell cycle control, they all cluster tightly together on the map.
So if you have an unknown gene, and it clusters with all the mitochondrial genes,
you have a powerful, immediate hypothesis that your unknown gene has something to do with mitochondrial function. It's a fantastic guide for future molecular studies.
Okay, let's move from analyzing existing mutations to the practical side of actually manipulating DNA.
Yeah.
We're getting into the core of recombinant DNA technology.
Right. To isolate a specific gene, you first need tools to precisely cut these massive genomic DNA molecules into pieces you can actually work with, and then you need to paste them back together.
The cutting tools are the restriction enzymes, right?
Right.
The restriction endonucleases.
Exactly, and they come from bacteria. They're these incredible molecular scissors that recognize very specific short sequences, usually 4 to 8 base pairs long, that are typically palindromic.
What's their natural job in bacteria?
They're part of the bacterial immune system. The goal is to chop up invading foreign DNA, like from a virus.
But how do they avoid chopping up their own DNA?
That's the other half of the system. The bacteria also make modification enzymes that add methyl groups to their own DNA at those same recognition sites. That methylation blocks the restriction enzyme, protecting the host's genome. It's a really elegant defense mechanism that we've just co-opted for the lab.
And many of these enzymes, like the famous E-Cori, make these staggered cuts, which we can see in Figure 6.11. Why is that staggered cut so important for cloning?
Staggered cuts create these single-stranded overhangs that we call sticky ends. And this is really the molecular basis of recombinant DNA. If you cut your gene of interest and your vector DNA with the same enzyme, they'll both have matching sticky ends.
And because they're complementary, they can transiently base pair together.
Exactly. They'll anneal. But that's a weak interaction. You need to seal them permanently.
And that's the job of DNA ligases.
Right. An enzyme like T4 DNA ligase, as shown in Figure 6.12, comes in and covalently joins the backbones of the aligned sticky ends. That seals the deal, creating a stable, circular recombinant DNA molecule.
So now we have a single functional recombinant molecule,
Yeah.
but we need billions of copies to do anything with it. We need to amplify it.
Which is where our bacterial workhorse comes in, the E. coli plasmid vector.
So plasmids are these small, circular pieces of DNA naturally found in bacteria, right?
Yeah, and they replicate independently of the main chromosome. We've engineered them specifically for cloning, as you can see in Figure 6.13. A good cloning vector has to have 3 essential parts.
Part 1, the replication origin, or ORI.
That's the sequence that tells the host cell's enzymes, hey, start copying here. It ensures the plasmid gets replicated every time the cell divides.
Part 2, the selectable marker.
This is usually a drug resistance gene, like AMP for ampicillin resistance. This is absolutely necessary because the process of getting the plasmid into the bacteria, called transformation, is incredibly inefficient. You need a way to select for the tiny fraction of cells that actually took it up.
And the third part is the polylinker, or multiple cloning site.
This is a synthetic region we have engineered to be packed with a whole bunch of unique restriction sites. It gives you a lot of flexibility for inserting your DNA fragment without accidentally cutting somewhere else in the vector.
So the transformation process itself, shown in figure 614, is just about getting that DNA into the E. coli.
We basically shock the cells with heat and calcium chloride to make them temporarily permeable. Maybe only 1 in 10,000 cells will actually take up the plasmid.
But then you grow them on a plate with ampicillin.
And only the transformed cells survive. They multiply and form a colony, a clone, and in the process they amplify your DNA fragment millions of times over.
Plasmids are great for fragments up to maybe 20 kilobases. But for mapping big genomes, like the human genome, we needed to carry much larger sequences.
For massive fragments, up to 2 megabases, we use things like bacterial artificial chromosomes, or BACs. They're derived from a different replication system in E. coli, and they're kept at a very low copy number, which makes them very stable. They can reliably carry huge chunks of DNA without getting messed up.
Okay, so once we can clone fragments, the next logical step is to build a comprehensive collection of them, a DNA library. And there are two main types: genomic and cDNA. Let's start with the genomic library in yeast. How do we use one to find a gene?
A genomic library contains clones that represent every single sequence in an organism's genome. To find a specific gene, especially one we've identified through a mutation, we use a technique called functional complementation.
And for that, we need a special tool called a shuttle vector. Why a shuttle?
Because you need to build and amplify the library in E. coli, which is fast and easy, but then you need to test the function of the genes in yeast. So the shuttle vector, as shown in figure 615, has components for both hosts: a bacterial origin and marker, and a yeast origin, centromere, and selectable marker.
Let's walk through the example of isolating the wild-type CDC28 gene, which is in figure 616. We start with a yeast strain that's a double mutant. It's temperature sensitive for CDC28, and it also can't make uracil because of the URA3 mutation.
Right. So first you transform that yeast strain with your genomic library. Then you select for the cells that successfully took up any plasmid by growing them at the permissive temperature, 23 degrees C, on a medium that lacks uracil.
So only cells that got a plasmid, which carries the functional URA3 gene, will survive.
Exactly. Now you have thousands of colonies, each with a different plasmid from the library, but they all still have that bad CDC28 gene in their genome.
Now comes the screening step.
You replica plate those surviving colonies onto a new plate and stick it at 36 degrees C, the non-permissive temperature. Now, only the rare colony that happened to pick up a plasmid containing the wild-type CDC28 gene will be able to grow.
Because that plasmid gene complements the defect in the genome.
And that's how you find it. You've isolated the clone with your wild-type gene, which you can then recover and sequence.
Functional complementation is great, but genomic libraries for higher eukaryotes are just unwieldy. The genomes are huge and they're full of non-coding introns.
Which is exactly why we need cDNA libraries.
So cDNA is complementary DNA, a copy of messenger RNA.
Right, as shown in figure 617. And since mRNA only represents the expressed protein coding parts of genes, and it's had all the introns spliced out, a cDNA library is way smaller and much more direct for finding a specific coding sequence.
How do you actually make these DNA copies from RNA?
You start by isolating the total mRNA. Eukaryotic mRNA has a unique feature, a long poly A tail at its 3-foot end.
The string of adenines.
Yeah. And we use that to grab it. We use complementary oligo-DTs bound to a matrix. Then we add an enzyme called reverse transcriptase, which was originally discovered in retroviruses.
And that enzyme uses the mRNA as a template to synthesize a DNA strand.
Exactly. Starting from that oligo-DT primer, that gives you a DNA-RNA hybrid, which you then process to make it double-stranded cDNA. You add synthetic linkers with restriction sites on the ends, cut them, and ligate the whole thing into a plasmid vector.
What are the challenges here?
A big one is representation. Genes that are highly expressed, like actin, will be massively overrepresented in the library, making it really hard to find transcripts from rarely expressed genes. For a mammalian library, you might need 10 million clones just to have a decent shot at finding one of those rare ones.
Libraries were the old way. The modern way to grab a specific piece of DNA, if you know the sequence at its ends, is the polymerase chain reaction, or PCR.
Oh, PCR is the bedrock of modern molecular biology. It lets you exponentially amplify one specific sequence out of a highly complex mixture.
And it's a beautifully simple three-step cycle, as you can see in Figure 618, just repeated over and over.
Yep. Step 1, denaturation. You heat everything to about 95 degrees C to melt the DNA and separate the two strands.
Step 2, annealing.
You cool it down to 50-60 degrees C. This lets your custom-made oligonucleotide primers, short pieces of DNA complementary to the ends of your target sequence, hybridize to the single strands.
And step 3, elongation.
You raise the temperature to 72 degrees C, the optimal temperature for your heat-resistant DNA polymerase, usually Taq polymerase, to get to work. It synthesizes new DNA strands starting from those primers.
And the magic is that in each cycle, you're doubling the amount of DNA that lies between the two primers.
Which leads to exponential amplification.
Oh.
After just 20 cycles, you have a million-fold more of your target sequence than anything else in the tube.
And for cloning, this is amazing.
It is. As shown in Figure 619, you can design your primers to include specific restriction sites. So the amplified piece of DNA already has the right sticky ends built in, ready to be cut and pasted directly into a vector.
And of course, there's RT-PCR for looking at gene expression.
Reverse transcriptase PCR. You just combine the first step of making cDNA from mRNA with the amplification power of PCR. It lets you amplify specific cDNAs from a tiny amount of starting material. And when you do it quantitatively, qRT-PCR, it's one of the most accurate ways we have to measure how much of a specific mRNA is in a cell.
Okay, let's talk about sequencing. We move past the old methods to next-generation sequencing to handle these massive data sets.
Right. The new methods allow for millions of sequencing reactions to happen at the same time on a solid surface. In one popular method, you amplify billions of short DNA fragments in place, and then you use super-sensitive cameras to watch as fluorescently labeled nucleotides are added one base at a time.
Generating billions of short sequence reads in a single run.
Exactly. And then, to assemble an entire genome, you use a computational approach called whole genome shotgun sequencing. You just sequence random clones over and over until you have, say, 10 times coverage for every segment.
And then massive computer algorithms just look for all the overlapping pieces and stitch them together to reconstruct the whole genome, as shown in Figure 622.
It completely bypasses the old, slow method of physically ordering all the clones first.
So having all of these genome sequences, human, mouse, yeast, you name it, is the raw material for the field of bioinformatics.
Absolutely. A field dedicated to extracting biological meaning from these massive, massive data sets, turning sequences into functional insight.
What's the core principle of bioinformatics?
Shared ancestry. All life comes from common roots. So if you see a significant sequence similarity or homology between two genes, it means one of two things. Either they have a recent common ancestor or, more importantly, that sequence has been conserved.
And conservation is just molecular evidence of selective pressure.
Exactly. If a sequence has survived for 1 billion years, from yeast to humans without changing much, it must be doing something absolutely critical. Natural selection has gotten rid of any bad variants. The core principle is the more conserved a sequence is, the more important its function is likely to be.
How does a computer use this to find genes in a new genome? It sounds straightforward for something like yeast, but really hard for humans.
For prokaryotes and simple eukaryotes like yeast, it's pretty easy. You just scan for open reading frames, or ORFs, long stretches of codons without a stop codon. Statistically, an ORF longer than about 100 codons is almost certainly a real gene.
But that trick doesn't work for us.
Not at all. In higher eukaryotes, our genes are chopped up into multiple short exons separated by long introns. The average human gene has 9 introns, so simple ORF scanning is useless.
So you need more sophisticated algorithms.
Right. They combine a ton of data. They look for matches to known cDNAs or partial sequences called ESTs. They use models to predict where the splice sites are, and critically, they compare the sequence to other species, like the mouse.
Because the non-functional DNA will have diverged a lot between humans and mice, but the important parts, the exons and the splice sites, will still be highly similar.
That conservation is a huge red flag for a functional gene. And once you've identified a gene, comparing a mutant sequence to the wild type lets you predict what will happen to the protein.
A total loss of function, a null allele, usually comes from something dramatic like a nonsense mutation, a premature stop codon, or a frame shift.
Right. Whereas missense mutations, which just swap one amino acid for another, are trickier. The severity depends on how chemically different the new amino acid is and whether that specific position is highly conserved across species. Changing a highly conserved residue is likely to be disastrous.
And for deducing function, the real power comes from searching all the known databases. The standard tool for that is BLAST.
Basic local alignment search tool. You give a computer your protein sequence, and it compares it against the entire database of known proteins, calculating alignment scores.
And it ranks them by a p-value.
The p-value is the probability of finding that degree of similarity just by random chance. A p-value less than 1 in a million, 10 to the $6, is generally considered solid proof of common ancestry, and therefore likely a similar function.
The story of the human NF1 gene is the absolute gold standard for this kind of deduction.
Oh, it's a perfect case study, shown in Figure 623. So mutations in the NF1 gene cause neurofibromatosis 1, a disorder with lots of tumors. When the NF1 protein was sequenced and run through BLAST, a big part of it showed profound homology to a yeast protein called IRA.
And what did we already know about IRA in yeast?
We knew IRA was a GTPase activating protein, a GAP. Its job was to negatively regulate RAS, a major protein that controls cell division and growth.
So the sequence similarity to IRA gave an immediate hypothesis for what NF1 does in humans.
Instantly. It suggested NF1 must also be a RAS regulator, and functional studies immediately confirmed it. NF1 is a RAS GP, so a defective NF1 protein can't shut off RAS, leading to abnormally high RAS signaling, which drives the excessive cell division in the tumors.
And that whole disease mechanism was predicted just because a human protein sequence matched a yeast protein sequence.
That's the power of it. And even if you don't find a global match with BLAST, you can still get clues by searching for smaller structural motifs.
Like a zinc finger or a kinase domain.
Right. These are short, recurring segments that do specific jobs. Motif databases can find them even if the rest of the protein is totally different, giving you partial functional clues.
Sequence analysis also lets us define evolutionary relationships really clearly. What's the difference between a paralog and an ortholog?
They're both types of homologs, meaning they derive from a common ancestral sequence, as illustrated in Figure 624. Paralogs come from a gene duplication event within the same species lineage.
So like alpha-tubulin and beta-tubulin.
Perfect example. They arose from a duplication of an ancestral tubulin gene, and now they're paralogs that work together to form the microtubule building block.
And orthologs.
Orthologs result from speciation. Human alpha-tubulin and yeast alpha-tubulin are orthologs. They diverged when the human and yeast lineages split. Orthologs are the most likely to have the exact same function in different species.
Okay, but the full genomic inventory revealed probably the biggest surprise of all, the complexity paradox. A human is just astronomically more complex than a roundworm, C. elegans, yet we have roughly the same number of protein coding genes, around 21,000 for us, 20,000 for the worm. Why?
This realization just fundamentally shifted molecular biology. It proved that complexity doesn't come from the number of parts, but from the complexity of their regulation. We figured out there are 3 major reasons for this paradox, shown in Figure 625.
Number 1, alternative splicing.
Right. In humans, a single gene can produce multiple different versions of a protein by splicing the exons together in different ways. The average human gene makes about 6 different variants. So our 21,000 genes generate a much larger, more diverse set of proteins than the 20,000 genes of the worm.
So we have fewer parts, but each part is designed to do many more things.
Exactly. Number 2, post-translational modification. Proteins get chemically altered after they're made. Phosphorylation, glycosylation, all sorts of things. These modifications create huge functional diversity that isn't directly encoded in the gene sequence.
Then number 3, the hardest to study, epigenetic effects.
These are stable, heritable changes that are not based on the DNA sequence itself, things like changes to chromatin structure. This creates this fantastically complex layer of control over when and where those 6 splice variants get made and how they get modified. It's this regulatory complexity, not the size of the parts catalog, that makes us different from a simple worm.
Now, applying these powerful techniques to human genetics is a lot more complicated. We can't do controlled crosses. We have to rely on just observing inheritance patterns in families to find disease genes.
We classify these single gene or monogenic diseases into 3 main patterns, which are in Figure 626. Autosomal dominant traits like Huntington's only require 1 bad copy. And the molecular defect there, a CAG repeat expansion, clearly implies a gain of function. The mutant protein aggregates and does active damage.
And you have autosomal recessive diseases like cystic fibrosis, which require two bad copies.
Right, so the parents are almost always heterozygous carriers. CF is caused by a defective chloride channel, and the recessive nature tells you it's a loss of function. One good copy is enough for normal function, so you only see the disease when both are broken.
And finally, X-linked recessive disorders like Duchenne muscular dystrophy.
These are on the X chromosome, so they show up overwhelmingly in males who only have one X. They can't mask the recessive defect. Females have a second X, which usually protects them, so they can be asymptomatic carriers.
So once you know the inheritance pattern, you still have to find the gene, and that's done through genetic mapping.
Which is based on recombination frequency. The principle, shown in figure 627, is that during meiosis, chromosomes cross over and recombine. If a disease gene and a known molecular marker are physically close together on a chromosome, if they're linked, recombination between them is rare.
So they tend to be inherited together.
Exactly. The genetic distance is measured in centimorgans, where 1 centimorgan equals a 1% recombination frequency.
And since we don't have enough visible markers in humans, we use molecular markers to track this.
The main ones are single nucleotide polymorphisms, or SNPs, single base differences, and short tandem repeats, STRs, which are short repeating sequences. STRs are great for family studies because they're highly variable in length, which makes the inheritance patterns really easy to trace.
But even with a lot of family data, this linkage mapping usually only narrows down the gene to a region of about 1 centimorgan, which is still a megabase of DNA. That could have 10 candidate genes in it.
It could, as shown in figure 628. So to find the one right gene, you have to switch from the genetic map to the physical map. First, you might do expression analysis. Look at those 10 genes and see if one of them is missing or altered in the affected tissue.
And second, and this is the definitive step, is sequence analysis.
You sequence that whole 1 megabase region in affected families, and you hunt for a mutation, a nonsense, a frame shift, something really damaging that is always present in people with the disease and absent in those without.
And this gets way harder when we talk about complex traits. Common diseases like heart disease or diabetes, they're caused by multiple genes plus the environment.
For those, the tool is the Genome-Wide Association Study, or GDOI.
Right. And with GDOIs, you're not looking at families anymore.
No, you're comparing the SNP profiles of a massive group of cases, say 5,000 people with type 2 diabetes, against a matched control group. You're looking for statistical correlations between common SNPs and the disease. The goal isn't to find the gene itself, but the region that's associated with the trait.
But how can these studies, which only track common SNPs, find what are likely rare disease-causing mutations?
They rely on a concept called linkage disequilibrium, which is shown in figure 629. When a new rare disease mutation arises, it happens on a specific ancestral chromosome that has a particular pattern of common SNPs, a haplotype.
Okay.
Over many generations, recombination slowly breaks that association down, but it's a slow process. So for a long time, the rare disease allele remains statistically associated with those neighboring common SNPs.
So GWSS finds the common SNP, which acts like a flag pointing to the real rare culprit lurking nearby on that same ancestral haplotype.
That's it. It lets you map the region without having to sequence every single person from scratch.
And these studies have also been incredible at finding genes that protect against disease, not just cause it. The story of PCSK9 is a huge pharmaceutical success that came from this.
It's a fantastic example. PCSK9 was first found because some dominant gain-of-function mutations caused high LDL cholesterol. So the critical hypothesis was, okay, if a gain-of-function increases LDL, then a loss of function should decrease it.
A pretty logical next step.
And they went looking, and sequencing confirmed it. They found people with natural frameshift or nonsense mutations in PCSK9 who had dramatically lower levels of LDL cholesterol.
So that validated the idea.
It totally validated it. It proved that PCSK9 normally act to get rid of the LDL receptor. So if you inactivate PCSK9, the receptors stay active longer and clear more cholesterol from the blood. This discovery directly led to the development of PCSK9 inhibitor drugs, a whole new class of powerful cholesterol-lowering therapies.
Finally, let's touch on cancer genomics. Sequencing a cancer cell reveals thousands of mutations. How do you sort through that noise?
You have to distinguish between driver mutations and passenger mutations. Driver mutations are the ones that actually contribute to the cancer. You can spot them because they show up repeatedly across many different tumors and usually cause severe functional changes. Passenger mutations are just random collateral damage, often silent, and don't contribute to the disease. Sifting through thousands of cancer genomes lets you find those recurrent driver patterns, which tells you which genes to target with therapy.
Okay, so we've established what a gene is and how to find it. Now we need to know where and when that gene is actually turned on. The most fundamental technique for this is in situ hybridization.
Right. In situ, as shown in figure 630, is all about location. It uses the specificity of base pairing, but you do it directly on a fixed, intact piece of tissue. You use a complementary labeled probe, often with the fluorescent tag, to find your target mRNA right inside the cells.
And the key advantage is that it preserves all the spatial information.
Exactly. It lets you map the precise location of gene expression within an embryo or an organ. It's how we know exactly where a gene like Sonic Hedgehog is expressed in a mouse embryo, in the notochord and the floor plate, which gives us huge clues about its role in development.
So in situ gives you a location for one gene. But to understand the whole system, you need to look at thousands of genes at once. That's where DNA microarrays come in.
Microarrays are all about high-throughput hybridization. You spot thousands of different gene-specific DNA sequences onto a glass slide, creating a DNA chip.
Let's walk through that classic two-color experiment in figure 631.
Okay, you take mRNA from two conditions, say fibroblasts treated with serum and untreated fibroblasts. You convert the control mRNA to cDNA and label it green. You convert the experimental mRNA to cDNA and label it red.
Then you mix them together and wash them over the microarray chip.
Right, and the color at each spot tells you the expression ratio. If a spot is yellow, it means equal amounts of red and green bound, so the gene's expression didn't change.
If it's bright red,
The gene was strongly induced by the serum. If it's green, it was repressed. In one shot, you can see which of thousands of genes responded to your treatment.
but the real power comes from analyzing data from many experiments using cluster analysis.
Exactly. If you look at expression profiles over time or across many conditions, as in figure 632, computer algorithms can group together genes that show similar patterns of regulation.
So if a bunch of unknown genes all turn on at the same time as known cell cycle genes,
You can infer that they are probably also involved in the cell cycle. The cluster map becomes a powerful predictive tool, grouping genes into pathways and giving you strong hypotheses about their function based purely on when they're turned on and off.
microarrays were revolutionary, but they did struggle with sensitivity. The next generation of technology moved to direct sequencing, RNA-Seq.
RNA-Seq has a vastly greater dynamic range, maybe 100,000-fold better. Instead of measuring hybridization signals, you just directly sequence a massive number of cDNAs. The abundance of each mRNA is determined by simply counting how many sequence reads match that transcript. It's a much more direct and sensitive measurement.
And the refinement of that, single-cell RNA-Seq, has been transformative.
It has, because it measures RNA abundance in individual cells. Bulk analysis averages everything together, which can hide critical differences between cell types in a complex tissue. Say RNA-Seq gives you that cell-by-cell resolution you need to understand, say, all the different kinds of neurons or immune cells in a single sample.
Shifting gears a bit, a huge application of cloning is turning organisms into protein factories, especially for therapeutics like insulin.
For that, we often use engineered E. coli expression systems, which are in figure 623. You clone the cDNA for your protein downstream of a strong inducible promoter. You add an inducer, like IPTG, and it flips a switch, telling the bacteria to start churning out your protein at a massive rate.
And to make purification easier, you often add a tag.
A standard trick is to add a short sequence of 6 histidine residues, a his tag, to the protein. That his tag protein will then bind super tightly to a nickel column, which lets you purify it away from all the thousands of native E. coli proteins in one easy step.
But bacteria can't always do the complex modifications needed for mammalian proteins, like glycosylation.
For those, you need eukaryotic expression systems, using cultured animal cells. You introduce the vectors via transfection. If you do a transient transfection, as in figure 634a, the plasmids replicate rapidly but are eventually lost, so you get high but temporary expression.
But for long-term experiments, you need stable transfection.
Right. In that case, the vector integrates randomly into the host cell's genome, as shown in figure 634b. You select for these rare events with an antibiotic, and the cells that survive have permanently integrated your gene and will express it forever.
And to make that stable integration really efficient, You can use viral systems like lentiviruses.
Engineered lentiviruses, shown in figure 635, are incredibly efficient. They use their natural machinery to infect the cells and integrate the gene of interest into the host chromosome. You get stable expression in virtually every cell.
Finally, we need ways to actually see what's happening inside the cell, using gene and protein tagging.
The most famous reporter for this is green fluorescent protein, or GFP, but it's really critical to distinguish between two tagging strategies, shown in figure 636. A promoter fusion links the gene's regulatory region to GFP. This shows you where and when the gene is turned on, because the whole cell will glow green.
But if you want to know where the protein itself goes,
Then you need a protein fusion. You link the GFP coding sequence directly to your gene's coding sequence. This reveals the cellular localization of the protein. You can see if it's in the nucleus, the mitochondria, or in one example, targeted specifically to the cilia of a neuron.
and beyond fluorescence, there's bioluminescence, using the luciferase enzyme from fireflies.
Bioluminescence is amazing for tracking cells in vivo in a whole animal. You can label T cells with a luciferase reporter and then actually image the light they produce through the skin of a living mouse. It lets you watch where they go in real time, which is just impossible with a standard microscope.
So we started by studying natural mutations. Let's conclude with the ultimate goal: actively designing and engineering precise changes to the genome ourselves.
This is the toolkit of gene function by design. And we start with yeast, which is still the gold standard because it has this incredibly robust homologous recombination system. It's very efficient at swapping out DNA sequences.
Which allows for precise gene disruption in yeast, as shown in figure 637.
You use PCR to create a DNA construct that has a selectable marker, like for G418 resistance, flanked by short sequences that match your target gene. You introduce that into a diploid yeast cell, and homologous recombination will replace one copy of the target gene with your marker.
And the analysis is simple.
If the gene is essential, when that diploid sporulates to make haploid spores, half of them will be dead. It's a clear confirmation of essentiality. This strategy was used to systematically knock out all 6,000 yeast genes.
The real innovation, though, was adding molecular barcodes.
To each of those deletion strains, they added a unique 20 nucleotide sequence of barcode. This allows for parallel screening. Instead of testing 4,500 mutants one by one for sensitivity to salt stress, you just pull them all together, apply the stress, and then sequence the barcodes from the surviving population.
And if a strain's barcode becomes rare, it means that strain was less fit. Its gene was important for dealing with salt stress.
It's an incredibly efficient way to define gene function in vivo, and that drive for efficiency is what led to the most revolutionary gene editing tool of our time, CRISPR-Cas9.
CRISPR is adapted from a bacterial immune system, but it's been repurposed for surgical genome editing in almost any organism. It has two core components, right?
Right, as you can see in figure 638. First, you have the Cas9 nuclease, which is the scissors that cuts the DNA. And second, you have an engineered single-guide RNA, or gRNA.
And the gRNA is the GPS.
It is. It has a part that binds to Cas9 and a 20 nucleotide target sequence that base pairs perfectly with the spot in the genome you want to edit. It directs the Cas9 scissors to that precise location where Cas9 makes a double-strand break.
And the cell then tries to repair that break, which gives you two possible outcomes.
The default pathway is non-homologous end joining, or NHEJ. It's error-prone. It often chews away or adds a few bases when it sticks the ends back together. If that happens in the middle of a gene, you get a frame shift, which creates a null allele, a permanent gene knockout.
But for precise editing, you need the second pathway.
That's homology-directed repair, or HDR. For this, you supply the cell with a DNA template that contains the exact change you want to make. The cell can then use that template to do a perfect, error-free repair. This is how you can do surgical editing, like correcting the single base mutation that causes cataracts in mice.
And just like the yeast barcodes, CRISPR is now enabling genome-wide screens in mammalian cells.
You can pool thousands of different gRNAs, each targeting a different gene, and introduce them all at once. If you're screening for resistance to a drug, the cells that survive will be enriched for gRNAs that knocked out genes required for the drug's action. You just sequence the gRNAs to find all the hits.
What if you don't want a permanent knockout? What if you just want to turn a gene up or down for a little while?
Then you use inducible CRISPR systems, laid out in figure 639. These use a nucleus-dead Cas9, or dCas9, that can bind to DNA but can't cut it. In CRISPR interference, or CRISPR, the gRNA directs dCas9 to a gene's promoter, where it just sits there and physically blocks transcription. It represses the gene.
And for activation.
For CRISPR activation, or CRISPR, you fuse dCas9 to a transcriptional activator domain. The gRNA brings this fusion protein to the promoter, and it transiently turns on transcription. It lets you study gain-of-function phenotypes without making any permanent changes to the genome.
For complex organisms like mice, knocking out an essential gene is often lethal to the embryo. To study its role in a specific tissue, you need a conditional knockout.
The LOX-P-Cre recombination system is the elegant solution for that, shown in Figure 640. It lets you inactivate a gene only in certain cells or at certain times.
And it requires making 2 different mouse lines.
Correct. First, you make a mouse line where your target gene is flanked by 2 special recombination sites called LOX-P sites. The gene is floxed. Second, you make another mouse line that expresses the Cre recombinase enzyme, but only under the control of a tissue-specific promoter.
Then you mate the 2 mice.
And in the offspring, the Cre protein will only be made in the cells where that specific promoter is active. And only in those cells will Cre recognize the LOX-P sites and cut out the DNA between them, disrupting the gene.
So you could, for example, knock out a receptor gene only in the hippocampus to study its role in memory without affecting the rest of the mouse.
It's an incredibly powerful way to study essential genes in specialized contexts.
Finally, we have a method that doesn't touch the genome at all, but instead destroys the mRNA. RNA interference, or RNAi.
RNAi, which is in Figure 641, is a natural defense mechanism. The process starts when an enzyme called dicer finds double-stranded RNA and chops it up into short interfering RNAs, or siRNAs.
And those siRNAs then get loaded into the RIC complex.
Right, and RISC uses the siRNA as a guide to find the complementary target mRNA, which it then cleaves and destroys. A small amount of dsRNA can trigger the silencing of thousands of corresponding mRNAs.
So it's a great tool for rapid, large-scale functional screens.
It is. Researchers used it to functionally inactivate over 86% of the genes in C. elegans just by injecting dsRNA, and they found over 1,700 new, visibly abnormal phenotypes, giving this unprecedented functional map of the worm's genome.
So what does this all mean? We've covered this huge molecular genetic toolkit, from classical mutation analysis all the way to the revolutionary precision of CRISPR.
Yeah, these techniques allow us not just to observe the cell, but to actively manipulate it, to map cause and effect right down to the single nucleotide. We're systematically deciphering the fundamental rules of life.
Let's just quickly consolidate the key takeaways from this journey of molecular control.
Okay. First, the fundamental insight you get from just studying a mutation, whether it's recessive, which is often loss of function, or dominant, often gain of function, gives you immediate functional clues. And using genetic interactions, especially synthetic lethality, is just indispensable for finding the redundant pathways that make cells so robust.
Second, DNA cloning with restriction enzymes and ligases is still crucial for isolating and amplifying genes, whether from a genomic library or, more practically, from a cDNA library. And PCR is the engine that drives all of it today.
Third, bioinformatics has shown us that conservation is the roadmap. Blast searches use evolution to deduce function, and mapping human diseases from linkage to GWAS uses statistical association to find causative risk factors.
And finally, the revolutionary ability to precisely engineer the genome, moving from homologous recombination in yeast to the surgical precision of CRISPR-Cas9. This lets us create targeted knockouts or precise edits while still being able to control expression temporarily or in specific tissues.
We've established that biological complexity, our complexity, doesn't come from the number of our protein-coding genes, but from the incredible complexity of their regulation.
Right, alternative splicing, post-translational modifications, epigenetics. And that leads us to a final provocative thought for you to chew on. We now have the tools, like single-cell RNA-Seq and genome-wide CRISPR screens, to analyze not just the protein coding sequences, but the vast, complex, non-coding regulatory regions of the genome. If the difference between a human and a roundworm lies primarily in that regulatory complexity, what further revolutionary insights about cell fate and development might we uncover when we finally finish writing the full regulatory manual of the cell?